Hello, my name is Maria Prieto. I'm going to talk about the Lichinomycetes, about a phylogenetic relationship within the class, generic and family delimitation, and the evolution of characters traditionally used to delineate these groups. So I have been, for this study, I collaborate with Martin Vesberg from Sweden and Matthias Schulz from Germany. Well, the Lichinomycetes is a class of lichenized Ascomycota comprising around 350 species distributed worldwide. They only contain only one order, and although there are very few species, they are very poorly known. Well, they grow on rocks and soil in moist or dry but temporarily wet localities, but they prefer to live in arid places and are pioneers on inhospitable substrates. So they present an importance ecologically, and they are mainly forming biological soil crust. Well, they form gelatinous lichen symbiosis, although they not always or not all of them look gelatinous. They develop a wide variety of growth forms, as squamulous forms or fruticose forms. And the cyanobiont is mainly a cyanobacteria. And again, there is a wide variety of cyanobacteria involved in this symbiosis, from unicellular forms with different division types and belonging to different orders, to filamentous species mainly belonging to the order Nostocales. Well, within the Lichinomycetes, there are three types of ascomata depending on the ontogeny. Typical apothecia are formed with an spheroid tangle of generative hyphae with trichogenes, trichogones and trichogenes, but not all of them develop from a primordium. Pycnoascocarps are developed when the generative hyphae arise within a pycnidium. And talinocarps are formed when the generative hyphae arise directly within the somatic plectenchima. Most of the species have prototunicate asci with only one layer, non-amyloid, and passive spore dispersion, but some of them develop unitunicate rostrate asci with an active spore dispersal through this protruding rostrum. Most of the species has 8 spores. Also, some of them can be polysporic with a maximum of 64 spores, and the spores are simple and colorless. Well, the position within the Ascomycota has been inferred several times, and in previous studies has been placed as sister group of the Geoglossomycetes or as an independent lineage or unresolved. In the last studies, they are placed as sister group to the Coniosibomycete, that is a newly described class of lichenized Ascomycota, in which we can find mass 88 species with typical prototunicate asci. Well, the split between these two groups and these two classes is estimated around 260 million years ago, and the diversification of the Lichinomycetes around 175 million years ago. Well, despite many efforts have been done to circumscribe genera and families, most of the studies have been based on morphology and anatomy, and only 2 studies have been used molecular data. The first study was done in 2001, and they only used one gene, the nuclear SSU region, and they tested the monophily of the order, including several members of 3 of the families. In a later study, in 2003, the same authors published a new phylogeny based in the same gene region, and they tested family delimitation. And as a result, they synonymized the family Hepiaceae within the Lichinaceae, but kept the Peltulaceae as a different family. But This taxon sampling was very limited, so we wanted to broaden this study and to study the whole class and as much families and genera as we could. So we developed this study, and our main aims were to test the current family and generic delimitation, to contribute to the understanding of the evolution of characters in the group, and to propose a natural classification. And to answer these questions, we developed a phylogenetic study based on 3 molecular markers: mitochondrial SSU, MCM7, and RPV2 protein coding genes, that we analyzed by Bayesian and maximum likelihood analysis, including 90 species and 142 specimens. And we used Lichinomycetes as outgroup. We also studied the evolution of characters by ancestral state reconstruction with maximum parsimony methods. Well, currently, within the Lichinales, there are 3 or 4 families accepted depending on the authors. The first family, Hepiaceae, was previously synonymized within the Lichinaceae, but this inclusion has not been followed by several authors. Currently, it includes only 4 species of the genus Epia, and although they share some characteristics with the other 2 families, Lichinaceae and Peltulaceae, they differ by having a corticated thallus and a vertical arrangement of the cells. They also differ from the Peltulaceae by having a filamentous photobiont and prototunicate and 8 spores in the ascent. Glioepiaceae is very similar to Hepiaceae, but different because it has an acorticated thallus with a radial arrangement of the cells in the thallus. It differs from the first one because it has a unicellular photobiont, prototunicate assae, and 8 spores. It includes genus Glioepia, Gudelia, and Pseudopeltula. The Peltulaceae is a well-defined family with around 60 species of Peltula that are mainly squamulous species, but also crustous species, and they are characterized mainly because they have a polysporic assae and unitunicate rostrate. And the Lichinaceae is the biggest family, and it includes a wide variety of growth forms, type of photobiont, types of ascomata, etc., and the type is Lichina. Well, this is the resulting Bayesian tree, in which I have marked the supported nodes by both bootstrap maximum likelihood and posterior probabilities with thicker lines. And as we can see in the tree, the Hepiaceae is here, and it is closely related with Pyrenopsis, that is a genus within the Lichinaceae. The Glioepiaceae is closely related with the type of Lichinaceae and some other crustous species. The Peltulaceae is sister group to Pecania, that is another group of Lichinaceae. And the Lichinaceae is spread along the whole tree and mixed with the other 3 families. So based on these results, none of the 4 families as currently defined are supported. So we are working on, well, if we want to keep these families as currently defined, we should erect many genera to family level that will result in a high number of families, or the other option could be to subsume everything within the Lichinaceae that is also not desirable. So we are working in the way to classify these families, and based on these results, we can at least see 3 supported clades that will be recognized at some level, probably at family level, but I will call lineages in this talk. So in the first lineage, there is the type species of the Lichinaceae, Lichina, plus some other important groups within the Lichinaceae, as Anema, Pterygiopsis, Sorotichia, plus the 2 families Hepiaceae and Glioepiaceae. In lineage 2, we have the family Peltulaceae, together with other groups and genera from Lichinaceae, as Pyrenopsis, Cryptotele, or Filiscum. And in lineage 3, we have several members of the Lichinaceae, and they will constitute a new family too. So based on these results, we wanted to relate the morphology with the molecular data. So we tried to study the characters and the ancestral reconstruction of some of the most traditionally used characters in the group. So the first one, growth form. Although in the Lichinomycetes we can find all of them look gelatinous, they develop a wide variety of growth forms, as I said. And they include almost all the forms that are present in lichens, plus the filamentous that is almost exclusive in this group. The first growth forms, that is crustose species, those that are very closely attached to the substrate, are very common in the whole class. So with the ancestral state reconstruction, all the ancestors for the class and for the three lineages are reconstructed as crustose species. And we can find in many places in the tree along the three lineages. In lineage 1, we can find in the genus Metamelania, in Sorotichia, Pterygiopsis, or Pyrenopsis. In lineage 2, there is only one genus, Crustose, that is Gonohymenia. And in lineage 3, both Cryptotele and Pyrenopsis are crustose species, crustose groups. While squamulous forms are those that are formed by small squamules, in this group are mainly peltate, and ancestral state reconstruction show that there has been many changes to this growth form, as in the genus Floeopecania, Polia, Gloeoepia, Anema, or Epia. And in this lineage 1, this character is defining monophyletic groups. On the opposite, in lineage 2 and 3, this growth form is mixed with other growth forms, as in Peltula clade, here, Filiscum, or Lympholaema, thus not defining monophyletic groups. Foliose forms are those that are very loosely attached to the substrate. In this case, they are very small, less than 3 centimeters, and they are not very common in the whole class. And in lineage 1, very few representatives only can find Lympholaema polycarpon, or in the genus Thyrea. In lineage 2, this growth forms is mixed again with the other growth forms, and in lineage 3 is not present. Well, fruticose forms are those with a shrubby habit, and they have appeared several times in the lineage 1, as in the genus Digitothyrea, Sinalisa, and Lichina, and they also define monophyletic groups. However, again, in lineage 2 and 3, this growth form is mixed with the other growth forms, as in Pecania, Lympholaema, or Lichinella. And finally, the filamentous forms that are quite unusually, because the overall structure of the thallus is mainly formed by the photobiont and surrounded by small cells of the microbiont, is only present in lineage 1 and is defined in genera as in Salbrucknerella, FB, and Thermutis. Well, as I have explained, we can find 3 types of ascomata depending on the ontogeny: apothecia, typical apothecia, thalinocarps, and pycnoascocarps. In lineage 1, both pycnoascocarps and thalinocarps are present, and the ancestral state couldn't be reconstructed, maybe due to the high number of transitions. And in this case, pycnoascocarps are defining monophyletic groups in many cases, but in some others we can find genera with both types, pycnoascocarps and typical apothecia. In lineage 2, we can find one clade, defined by pycnoascocarps and at least 2 clades with thalinocarps. This is the only lineage that presents thalinocarps and the only one without typical apothecia. And finally, in lineage 3, there are 2 clades defined by the type of ascocarps, both formed by several genera, one with typical apothecia and the other one with pycnoascocarps. Well, the number of spores is not so changing in the group, so main ancestor for the whole class and for the 3 lineages is reconstructed at 8 spores. And in lineage 1, several groups with multisporic assae has appeared, forming a monophyletic group as in Floeopecania, Sinalisa, or Salbrucknerella, but there are some other groups in which we can find both types of assae, as in Pterygiopsis or Ephebe. In lineage 2 and lineage 3, this character is defining monophyletic group, except in the genus Philiscum. Well, the photobiome has been traditionally used to delineate genera and families, but it's not used at the moment. So we can find a high diversity of forms in the photobiomes, depending on the division, like several orders of unicellular forms and some other groups of filamentous species, mainly belonging to the Nostocales. So the term cocoid is used when the photobiome is not really known and it is observed as unicellular. So there are many groups, many species in which we don't know the photobiome at the moment. So we are sequencing the photobiomes and to include in our results. Well, in lineage 1, there is a high number of cocoid forms, as you can see. Also, we can find other gloeocapsoid unicellular forms and members also of the filamentous group as Nostoc, Cytonema, Ribulariaceae, Calotrix, or Stygonema. In lineage 2, we have also several groups with cocoid forms. It's for the first time present Myxosarcina genus, and there is also a clade defined by Nostoc. In lineage 3, we can find only unicellular forms, and those are related with the clades, with cocoid forms, Croococtidiosis, and gloeocapsoid forms. So if I haven't shown all the characters that I have studied, but if we take all these characters in combination to try to delineate these lineages and to try to find a solution for the family delimitation, I can find some conclusions. Well, in lineage 1, there is the one with highest diversity of growth forms, plus is the only one with filamentous growth forms, and in this case, this growth form is defining monophyletic groups. They also have pycnoascocarps and typical apothecia with a high number of transitions. We can find 8 spore or multisporic assae, and they also have the highest diversity of photobiomes. In lineage 2, we have a high diversity of growth forms too, but the growth form is not defining monophyletic groups. This is the only one group with thalinocarps and without typical apothecia. We also can find a group with 8 spore and multisporic assae, and several groups, very few of photobiomes are found. Finally, lineage 3 has the less diversity of growth forms, and in lineage 2, they are not defining monophyletic groups. They have both typical apothecia and pycnoascocarps defining monophyletic groups. We also have 8 spore and multisporic assae, and again, defining monophyletic groups, and we only can find unicellular form photobiomes. I will explain a little bit about the generic classification. So, well, as a resume, I have found a lot of non-monophyletic genera within the group, and one of the cases is the genus Sorotichia. that is characterized by this crustose growth form with typical apothecia and cocoid photobiont. So many species has been included in Sorotichia by these characters. So many of them will be excluded soon of this genus. So if we observe in our tree, within lineage 1, we have this clade that is formed with the type species of the genus Sorotichia murorum, that is very closely related with Sorotichia schereri. But here there is another clade with Sorotichia vermiculata, Sorotichia lutophila, and Sorotichia lugubris. So this group is very polyphyletic. Another case of highly polyphyletic genus is Pyrenopsis, that is a really nice genus, very beautiful, but with very small apothecia, almost close in all the species, and is characterized by pycnoascocarps and the photobiont with a red cover. In this case, we can find one clade here within lineage 1 that is not related with this second clade here. We can find another clade here and another clade in the lineage 3. So in this group, we also cannot place the type species. So we are trying to figure it out. So as conclusions, we can say that none of the families are supported. So we are working on the new definition and recircumscription of these families that probably will correspond to the lineages. Many genera are polyphyletic, so many of them will be divided and segregated. The phylogeny is being very useful to study this group, and the character evolution is also being very useful to delineate these groups. And it's very interesting to see that all the many of the characters are following different evolutive patterns within each lineage. So with all this data, we hope that we will publish a new classification of the Lichinomycetes soon. So I would like to thank the people that has been helping me in the field to break all these very hard rocks and to collect, and all of you for listening. Thanks.